So we're going to be talking about the effect of ultraviolet radiation on bacterial growth and also looking at some examples of fungi. So I'm going to start by sharing my screen to look at the effect of UV radiation. So this is the handout that's on lab for this week. So of course you already know about UV radiation, right? What kind of damage does UV cause? Remember, you have to talk. What kind of damage does UV cause?
It can cause a thymine dimer.
Right, thymine dimers, as we see here. And so the UV radiation excites two adjacent thymines, and they actually chemically bond to each other. And the problem is that when either DNA polymerase or RNA polymerase happens to come by, when it sees that thymine dimer, it does not see two thymines. And so the polymerase enzymes cannot add those two complementary adenine nucleotides. And so a mistake occurs. And what do we call that mistake?
A mutation.
A mutation. So is there a way for cells to correct the problem?
Yeah, can't they cut it out somehow?
Right, so all those organisms that evolved with exposure to UV radiation have DNA repair enzymes, which constantly scan the DNA molecules, the chromosomes, and if they find a thymine dimer, they cut it out and replace the two thymines. But remember, there's only just so much damage that can be corrected, and the thing about bacteria is that they can replicate and divide in as little as every 20 minutes. So if you can cause enough thymine dimers, the bacteria cannot repair them in time, and so they die. So we're going to study the effect of UV and the duration of the UV light, and also see if certain plastics have any benefit. So we have a little YouTube video to watch. It just sort of gives a simplified example of the kind of thing that we do in lab. And there's no talking except for the advertisements. Oops. Gosh. So we just saw an example of exposing the bacteria to different amounts of UV light. Of course, it would have been nice if they'd used a clean index card instead of the dirty one that they used. But we're going to look at how to expose plates of bacteria to 20 seconds and 2 minutes. The first two plates are exposed with the lid off. All right, so Petri plates have plastic lids, so we take the lid off so we can expose the plates of the surface directly to UV light. The third plate, plate C, is 2 minutes, but with the lid on. And the last plate is just exposed to the ordinary room lights, no UV, so that's our control. Now, before you can start to zap your bacteria, you must completely cover the entire surface of the agar plate using a swab. So just as was shown in the video is how we would have done it. And you notice how she swabbed the plate side to side, top to bottom, and then rotated and did the same thing and rotated. So that way you get complete coverage of the agar surface. So it's very important. And then instead of an index card, we actually do have paper cutouts, heavy cardboard cutouts of different shapes and things. It could be stars, they could be faces, they could be X's or E's. So we do have different paper cutouts that we put over top of the plates, and then the UV light shines down through the cutout portion. And then we have to incubate the plates. So here we see a setup, which is like how we do it. We are using Serratia marcescens, a gram-negative rod. But we're using the strain D1, which is the strain that produces the deep red pigment. And it's actually one of the unknowns that some of you had. But regular Serratia does not produce pigment. But the D1 strain does produce the red pigment, but not at 37 degrees Celsius. You need to be at 30 degrees or lower, like room temperature, in order for the bacteria to produce the red pigment. So it's an interesting example of temperature-dependent gene regulation. Right, because the Serratia marcescens D1 will not produce red pigment at 37, but it will produce pigment below 30 degrees, like at room temperature. So we use it because the red pigment is easier. You get to see where the bacteria are. Plus, we're also going to see something interesting that can happen to those bacteria after they're exposed to UV light. So here we see the star-shaped paper cutout, but in the results, we used an X cutout. So plate A had the lid off with the X paper cutout on top and was exposed for 20 seconds. Plate B had the lid off and the paper cutout on top and was exposed for 2 minutes. Plate C, we left the lid on the plate and put the paper cutout on top of the lid and exposed for 2 minutes. And plate D was our control. It was just exposed to the ambient light in the room, so no UV. And so we see some red growth on these plates. So looking at plate A and B, what can you say in general about UV exposure? So what did UV do to the bacteria?
It killed the bacteria in a really short amount of time.
Right, so we see that UV kills, right? So UV kills, even with just 20 seconds, we can see that UV kills, right? We have a clear zone in the center. And when we expose the bacteria to 2 minutes, so a lot longer exposure time, we see that even more cells are killed, right? So remember when we talked about control of microbial growth, and one of the factors that affects the antimicrobial treatment is exposure time. Longer is better. And so we've just confirmed that here. Exposing bacteria to more UV longer results in more killing. A lot more killing, right? And then plate C, we left the lid on. And Petri plates are made from a type of plastic called polystyrene, a very common cheap plastic. It holds up pretty well, but does it block UV? Does polystyrene block UV? Look at plate C. And what do you see on plate C? So let me put it this way. Would you wear sunglasses made out of polystyrene?
Because it could still go through.
Right, so we can still see an X there, right? Which means enough UV got through to kill most of the bacteria. So polystyrene blocks some UV, but not enough. And so you would not want to wear sunglasses made out of polystyrene, but your sunglasses are plastic, right? And if you wear glasses, like I do, you probably have plastic lenses. Should you be concerned? Should you go get your money back? I mean, they're plastic, aren't they?
Put like a protective coating or something.
Mm-hmm. Well, your prescription glasses have an anti-glare coating that you can pay for, which is a nuisance because all it does is scratch very easily. But that just helps to prevent reflections. It doesn't block UV, not the anti-glare coating. But you all have sunglasses, right? And they're made of plastic, right? Yes?
Yep.
But it's not polystyrene. It's polycarbonate. So polycarbonate plastic effectively blocks UV light. At least 99% of UV is blocked by polycarbonate. So all sunglasses are made with polycarbonate plastic. If you have prescription glasses and you opted for plastic, it's polycarbonate plastic. So your eyes are protected from UV. Now the thing about polycarbonate, it is just a plastic and it's a cheap plastic. So how much money should you be spending on sunglasses? Answer, not that much. A $10 pair from Walmart is just as good as a $100 pair from Nordstrom's because it's all polycarbonate plastic. And by law, it must block at least 99% of UV. So all sunglasses sold, the plastic ones, are made of polycarbonate. And so all of them block the same amount of UV. So stop wasting your money on expensive sunglasses. By law, they all have to block the same amount of UV. Whether they're $10 or $100 or $200, it's all the same cheap polycarbonate plastic. So how much did you spend on your sunglasses? I only pay about $12 or $15 for mine. So you learned something useful today, didn't you? Now, I see something interesting on plate C. If you look in the kill zone, you see colonies growing. So there were some bacteria that survived the treatment. But what color are they?
White?
Well, they're actually sort of non-pigmented, right? But they're not red. They're still Serratia marcescens, but they're not red. So what happened to those cells? So tell me, what happened to the colonies in the kill zone on plate C?
Did they become resistant?
No, not resistant. They were probably cells that were underneath several other cells, and so they were able to survive because they had some other cells protecting them. But they're not pigmented. So what happened to them? Nobody has any idea? Bryant, do you have an idea? Ivy? Rachel? Any ideas?
No, I can't think of anything.
What about you, Bryant? Talk to me. Ivy? Sarah? Ruby? Taylor? Nobody has any ideas? Maybe I should make that a question on lab exam too. So what could be going on plate C in the kill zone? We had survivors. Are they normal?
No, maybe because the colors for protection. got like burnt out from the light.
Well, you're kind of sort of on the right track. The gene to produce pigment got mutated. It's no longer functional. So those bacteria survived, but they're mutants. And they lost the ability to produce the red pigment because of mutations in that gene. So does that make sense?
Yeah.
So remember, UV does cause mutations, and too much UV will kill, but a little bit causes mutations. And we've just seen that here with our Serratia marcescens on the agar plate. So what do you think it does to your skin cells? Is it a good idea to get nice and tan every summer?
Probably not.
Definitely not, right? Because the reason why you tan is in response to damage caused by UV. So your skin cells, your DNA has to start being damaged before you start producing melanin. So being nice and tan just means you got a lot of sun exposure and you got damage. And that damage is cumulative. So you may not see the effect until you're in your 40s or 50s. If you're lucky, all you will see is just wrinkles. If you're unlucky, you may see skin cancer. So it's always best to wear sunscreen if you're going to be outside for a significant period of time, more than 15 minutes. Wear sunglasses or other glasses that block UV, a wide brimmed hat for protection. And the ladies especially always love to expose their shoulders. That's why you see all those freckles, all the sun damage on people's shoulders, because they expose them a lot, right, to the sun. So you want to make sure and use a lot of sunscreen, right, to protect. So any questions on our plates?
Professor,
Yes?
on plate C, did you say the gene that produce pigments are not mutated?
They are mutated.
Okay, thank you.
And then plate D was our controls. It's covered with red Serratia because it was just, exposed to the overhead lights in the classroom. So there's no UV there. Now, what about the paper? Did the paper protect the bacteria from UV?
Yes.
Yes, it did, right? But you can't wear paper sunglasses, right? Because then you can't see anything. So again, we've shown that UV kills even with short exposure times, although longer is better. UV causes mutations to occur. Polystyrene plastic is not good enough to protect against UV, but paper is, but we wear clothes which block the UV, but you can't wear that over your eyes. So you can use polycarbonate plastic instead, and that blocks UV. So any questions on the results? So what's the primary lethal effect of UV light? What does it cause?
Mutation.
Well, mutations because it's causing what kind of specific damage? What kind of specific DNA damage?
The dimer.
Right, thymine dimers. And that can lead to mutations or death. What was the purpose of exposing plate C, which had the lid on? Why did we leave the lid on?
to protect for protection.
Well, no, not for protection, because it really doesn't protect very well, right? But you work with polystyrene petri dishes all the time, and you're growing your cultures. Isn't it a good idea to know if the lid protects against UV? What if you work in a lab where you use a lot of UV? Or many fume hoods, the way they work is that when you're finished, you turn a UV light on in the fume hood and leave it on all night long. And so when you come back, the fume hood is nice and sterile. If you left some culture plates in there, you would have killed all your bacteria. So it's nice to know whether or not the polystyrene actually blocks UV to protect them, because if it doesn't, you may need to take some extra precautions to protect your cultures if they might be exposed to UV light. Number 3, what are pyrimidine dimers? So remember, purines and pyrimidines are the nucleotides, the A, T, G, C, and U. And so specifically, what are the thymine dimers? What actually happens to create a thymine dimer?
They cause cancer, skin
Well,
cancer.
bacteria don't do cancer. Right, so more generally, what do they do?
They kill bacteria cells.
Okay, be more general. So remember, what are thymine dimers, right? Two thymines that are side by side. They get excited by UV radiation and they chemically bond to each other. And so when the polymerase enzymes come along, they cannot read a thymine dimer as two thymines. So the polymerase enzymes cannot add the correct adenine nucleotides. And so a mutation occurs. A few mutations can be repaired, but too many cannot, and that will cause cell death. Well, number 4, we talked about how to repair UV damage, right? So how do cells repair UV damage?
By cutting out the dimers.
And how do they do that? Just in general? By an enzyme, right, which can cut out the thymine dimers and replace them. Yes. And the fancy name for that enzyme is a photolyase, which you don't need to know, but they're interesting enzymes that can repair this damage. So any questions about UV?
How come dimers don't form with the other nucleotides?
Well, they may with other pyrimidines, but it's mostly the thymine dimers, but they can also occur with cytosine, but it's mostly thymine dimers that are the problem, but they can also occur with cytosines. That's why, yeah?
No, okay, thanks.
Yeah, so that's why the term here, pyrimidine dimers, although we mostly talk about thymine dimers, but they can also occur with cytosine. It's just I think it's easier for the thymines to chemically bond to each other. Any other questions? I have to go back and open. So I just got to open up fungi. So we all see fungi and yeast.
Yeah, we can see it.
Yeah, so normally this would just be an observation type lab anyway, where you would look at some live cultures, which we're not going to look at here, but you also look at a bunch of prepared slides of the different types of fungi. And then the goal was just to be able to recognize the type of fungus when you see it. So we're going to be looking at some slides of Penicillium, Rhizopus, yeasts, and Aspergillus. So our first organism here is Penicillium. What's special about Penicillium? What's special about Penicillium?
Is that what makes penicillin?
Huh?
Is that what they use to make penicillin?
Yep. Right. So that's the fungus from which penicillin comes. Is Penicillium. And so what we're looking at on these slides are actually reproduction in the fungi. And so with Penicillium, it does form a nice fuzzy kind of green or grayish green colony. And you may have seen this if you've had some moldy bread, the green spots on the bread are Penicillium. Maybe you had a large block of cheese and you left it in the fridge too long after using it. And again, you see some green fuzzy colonies growing on the cheese. That is also Penicillium. If you examine the green part microscopically, what you see are these structures here, which are called conidiophores. And they look kind of like the end of a broom or maybe like a skeleton hand that's kind of knobby. And it does have little round dots on it, and those are spores. So we're going to talk about fungi in lecture on Wednesday, just a little short lecture. And when the fungal colonies start getting colorful, that normally means that they're producing spores. And fungal spores are for reproduction, not survival as with bacteria, but for reproduction. And so fungi produce a lot of them. And they're very light, just a little bit of air movement, and they take off and fly around. And if they land someplace which has adequate food and moisture, they can germinate. And what you see microscopically are these structures here, the conidiophores, which are the fuzzy green parts of the colony. They are very small. Even on our slides, it's not easy to find and focus these things because they are so tiny. But here we see some Penicillium conidiophores. Now, Aspergillus is different. This one's easy to find, and also sort of the prettiest looking one. And they look kind of like flowers in the microscope when they're stained. But the conidiophore for Aspergillus is a ball. So it's a ball that's covered with spores. And what they've done on the slides is take a slice of that ball and stain it. And so when you take a cross section of the ball, this is what it looks like. And all these little dots, those are all the spores. So just imagine this as being a ball covered with spores, and you slice it and look at a tiny thin slice, and this is what you see. And here's another example where most of the spores have all been released and are scattered all over the place. And that's just one little conidiophore. A colony produces thousands of conidiophores, and they're covered with thousands of spores. And so, again, spores are for reproduction. Most of the spores will never become anything. They will deteriorate and die. But a few of them will land someplace where they can start to germinate and grow. When it comes to Rhizopus, this one here, we're looking at a zygote. And of course, we all remember what a zygote is, don't we? What's a zygote? You've all heard of zygotes before. Where have you heard of zygotes?
Fertilized ovum.
What was that?
Fertilized ovum.
Right, a fertilized ovum or a fertilized egg, right? Because you all remember when a sperm fertilizes the egg, it becomes a zygote. And what can you tell me about a chromosome number of sperm and eggs, of the gametes? What can you tell me about them?
You're half.
Right, and what's the fancy word for half the total number of chromosomes?
Haploid.
Haploid, right? Haploid. And so when the haploid egg and the haploid sperm fuse together, then what do you call the number of chromosomes?
Diploid.
Right, diploid. So when it comes to fungi, there are no eggs and sperm, but most fungi, if not all fungi, live their lives as haploid organisms with only half the number of chromosomes, and they're perfectly happy that way. But sometimes they will get together and engage in sexual reproduction. And so half the chromosomes from the two, what they call opposite mating strands of the fungi, come together. And so the genes get exchanged and you temporarily form a diploid structure. In this case, with Rhizopus, the zygote is that diploid structure. So you had your two opposite mating strands, which came together to exchange genes and form that zygote, which is a temporary diploid structure. The zygote will actually go on to produce spores, but spores are always haploid. So they're always haploid. because they're going to germinate and form a haploid organism. So spores can be the result of meiosis, as is the case with the zygote, or they can be the result of mitosis. A haploid fungus can make haploid spores. So either mitosis or meiosis can be used, but the end result will be haploid spores. And the zygotes are big. I mean, this is not even a 400 times magnification, and you can still see how big they are. So they're huge. But this is an example of sexual reproduction in a fungus. And as we're going to learn in lecture, it's the sexual phase of a fungus that is used to classify that fungus. And if they can't find the sexual phase, then they stick it into this general category we'll talk about. But usually, eventually, they do discover the sexual phase, so they can then classify the actual fungus. Now, when it comes to yeasts, yeasts are also haploid. Or actually, yeast genetics can be complicated. We're not going to get into yeast genetics, but yeasts mostly reproduce by budding. And budding is a form of asexual reproduction. It's an unequal cell division. And unfortunately, this slide really doesn't have any good examples of yeast with buds. But what you would see is a little cell stuck onto a larger cell. And that's the bud that's forming. And the fancy word for the bud is blastospore. But we just call it a bud. And so when yeasts are happy, they bud like crazy as a way to reproduce. But it is possible for yeast to get together and exchange genes in sexual reproduction. And that's what we see here in these slides. So in these slides, the yeast cells are stained kind of blue or bluish green. And the spores inside are very dark. They look almost black. And you can see these black things inside. And so those are the spores. And technically, they're called ascospores, but they are a form of spore. And so yeasts will only get together and reproduce sexually if they're stressed, if the environment is deteriorating. Maybe it's too dry, or it's becoming polluted and toxic. And so the yeast cells will actually get together in sexual reproduction to produce spores. So why is it when the yeasts are happy, they just bud, but when they're stressed, they have sex? What's the benefit of sexual reproduction? So what's that magic word in biology? What's the benefit of sexual reproduction?
To produce genetic variation?
Right, to increase variation, right, to increase diversity. The idea is if conditions are wonderful, then the yeast just bud like crazy because they don't need any diversity. They can just clone themselves. But if the environment is deteriorating, if conditions are becoming unfavorable, they can sense that. And so they get together and exchange genes. And the idea is that their offspring, hopefully, will be better adapted to survive. And so that's why fungi get together and sometimes reproduce sexually, is because they want to mix genes because they sense the environment is changing or deteriorating. And so they're trying to increase diversity. But if conditions are wonderful, there's no need. You can just clone yourself. So does that make sense? Oh, that does not make sense?
Makes sense.
Yeah, it makes sense, right? So, well, like I said, sometimes it's difficult to find the sexual phase for a fungus, but when they do, then they can actually classify it correctly. So you should be able to recognize these different fungi when you see them. I deleted some things, so that's why this is not numbered correctly. But these other terms here, we'll talk more about them in lecture. But the hyphae are all the fuzzy filaments you see in a mold. If they're all clustered together to form a solid structure, we call that a mycelium. And when they're old enough or mature enough to start reproducing, they send up aerial hyphae, which are the reproductive hyphae, which produce the spores. Now, when it comes to differentiating yeasts from molds, molds are fuzzy. They are filamentous. Yeasts are slimy. So it's easy to distinguish a mold from a yeast. Molds are fuzzy and filamentous. Yeasts are slimy. So like I said, you've probably seen moldy bread before, right? It's got penicillium on it. And if it's just a few little specks of penicillium, all you have to do is just, pinch those out. It's just on the surface of the bread. It's not in the bread. Right? The bread's perfectly fine. Just, cut or pinch off those little specks of mold, and the rest of the bread is fine. Same thing with cheese. If you got a block of cheese, has a big old green colony of mold growing on it, just cut that off. It's only on the surface. The rest of the cheese is fine. It's not inside the cheese. I mean, what do you think people in the old days did? You think they'd throw away a whole loaf of bread because it's got a few specks of green mold on it? They could not afford to do that. The same with cheese. Just cut off the bad stuff. I mean, cheese has got a lot of bacteria and some mold in it anyway, right? Does anyone like blue cheese salad dressing? Anybody like blue cheese salad dressing? It's okay to
Yeah.
admit it. Yeah, well, those chunks of blue are mold. So you're eating mold. So, and that's how that works, right? If you don't do a good job of, drying or disinfecting your shower or shower curtain, you get some kind of pink slime growing on it, or maybe some black slime. Well, that's a yeast, right? Because you didn't keep it clean. And so slimy are yeasts and fuzzy are molds. So any questions on the fungi? I mean, yeasts are fungi, but I just titled this fungi and yeast. No questions?
Professor,
Yes, Christiana.
molds, you say we can just cut off the part, the fuzzy part, right?
Right, so molds grow on the surface of your food, so just cut that off, and the rest of the food is fine.
Uh, so they don't go in the food.
Not very deeply, no.
Okay.
Just on the surface.
Okay. And for yeast, for example, you made example of the one in the bathroom. Do they cause any
Uh-huh.
harm? Are they
Well, not with normal exposure to it, but you should disinfect, your shower curtains or your, tile. Just keeping everything nice and dry will stop any uh, yeast and molds from growing. It's just that when areas stay damp, That's what allows for yeast and molds to grow. So you need to make sure that they dry out completely. Right, so either squeegee your tiles or wipe them with a towel so you can get off most of the moisture, and then that way you won't have any problem with yeast or molds. Any other questions?
No, thank you.
Okay, your exams are graded and posted, so you can go take a look. Your average was much higher than the morning classes, so overall, good job.
Thank you.
So, no other questions? All right, then Wednesday, lecture is on fungi, and we're going to start talking about viruses in general, and then focus on the bacterial viruses. Next week, we'll get into animal viruses and eventually coronavirus. So that's it.